Hello, welcome to our STEM News broadcast of our free choice scientific reading during spring quarter, week 3 of 2022. We're STEMNewsNexus at UCLA, blog of preprints and new publications. So we're an undergraduate student organization at UCLA. We love research and we're just trying to squeeze in some free choice scientific reading. We're looking for faculty mentors and graduate student mentors. So here's our email if you're interested. And if you're an undergraduate, please check our Linktree for educational opportunities. And the purpose of our organization, again, is just to make scientific discoveries more accessible. It's definitely like drinking out of a water fountain to keep up with all this new science and our current material. So we just want to kind of redefine STEM news to be something more practical rather than for entertainment purposes. And yeah, so let's get started. So today I'm going to be talking about this preprint on centrin-specific protease 1, zinc, and metabolic stress. Here is the title and author, and I'm referencing the one published or posted on April 6th of 2022. And I've only read the first 4 pages for this discussion. Because that's all I had time for this week. So essentially what they're talking about is insulin secretion, and insulin secretion from the pancreas and insulin becomes too insensitive in type 2 diabetes. But there are obese people with normal blood sugar, and they are seem to have an upregulated insulin secretion with a greater amount of beta cells in their pancreas. So a lot of people who study metabolic disease look at the isolate mass of the pancreas, which is supposed to measure how many different types, how many cells are within the pancreas altogether. And then mice on a high fat diet, which is associated with obesity, also secrete more insulin, preceding an increase in isolate mass. And there is a lack of this compensation may contribute to type 2 diabetes pathogenesis and progression. And our previous researchers have been trying to investigate how this works. And there has been some information talking about the charge of the cell and calcium ions with some interacting with some sort of unknown electron acceptor to like participate in this mechanism. And there also has been research talking about the centrin-specific proteases, which are basically like phosphatases, but for a ubiquitin modifier-based mechanisms or in the cell, as opposed to like phosphate-based inorganic phosphate transfers between different proteins. It's like ubiquitin transfers between proteins. And SEM1 is a suspect pathway protein for this compensation mechanism. So these researchers are asking or want to know how may an upregulation insulin work given the information that's already available. Does it have to do with SENP1 and the redox state of the cell? So the data that they looked, or the arguments they're making from their results is that redox signaling is involved or is required for this compensation in high-fat diet mice, and that there is this residue cysteine 535 on SENP1 that's near the catalytic site that may be controlled by zinc 2+ ion and a redox sensing mechanism in order for it to be involved somehow in the pathway that upregulates insulin secretion. And a knockout of SNP1 and beta cells and other cell-specific markers in the pancreas show that SNP1 is required for the compensation mechanism. So they're arguing that from their results that zinc ion binding in a redox reaction on SNP1 activates SNP1 to do an acute upregulation of insulin secretion in beta cells of the pancreas. So I think that looking at this data, you can go and see this preprint and kind of make your own judgments on whether you think it's good and leave a comment and kind of participate more in the scientific community.
Hi everyone, today I am going to be talking about how tuberculosis infection affects the severity of COVID-19 in patients. As you can see above, this is the article that I read this week. And since tuberculosis is a widespread infection in third world countries, Scientists have questioned whether the immune response triggered by this serious respiratory infection might protect people from developing COVID-19. Scientists ran an experiment with mice and discovered that mice who were infected with tuberculosis showed no signs of COVID-19, likely because the pre-existing immune response to tuberculosis prevents the virus from proliferating into the lungs. Tuberculosis infection makes the lungs inhospitable to the COVID-19 virus in mice. The findings may also explain why countries tend to have high rates of COVID-19 or tuberculosis, but not both.
I hope everyone has a great week 4, and I hope to see you next week around 10:30 p.m. Pacific Standard Time.